So today's topic of discussion is detection of adulteration in mutton and chevon and the practical approaches. Since our childhood to till now, whenever we eat a meat in our plate, the topic of specifically when we eat chevon and which is popularly known as mutton among all of us, the debate remains whether really it is from goat or it is adulterated from sheep. So it is now very important for us to know how we can detect these things in using practical approaches. So adulteration refers to substitution of meat of a costly or superior variety with a cheaper variety, which may be of undesirable or inferior quality or from a different species. So this may be objectionable not only from the ground of health, but when it comes to India, there are various religious sentiments. For example, in Hindu religion, beef cannot be accepted by the people. Similarly, when it comes to Muslim religion, addition of pork it cannot be tolerated by people. So we have to take account of everything and economics. Those who are meat producers to get some illicit financial gains. Usually, chevon is rampantly adulterated by mutton, which is the sheep meat. So this is punishable under the Prevention of Food Adulteration Act 1954. So apart from adulteration between chevon and mutton, some other common adulteration such as horse flesh adulteration in beef, cat flesh adulteration in rabbit, and donkey flesh adulteration in venison. Venison is deer meat. So let's come to some basic nutritional properties of meat. So as we all know, meat is a very good source of protein, B vitamins, and meat is the meat and other foods of animal origins are exclusive source of B12 vitamin. Apart from that, meat is a good source of iron and it is a good source of zinc also. Specifically, heme iron, it is 50 to 60% in case of beef, lamb, and chicken, and 30 to 40% in case of pork, liver, and fish. So, if you are consuming 100 gram of cooked meat, it provides 100% recommended dietary allowance of calorie, 50% of protein, 35% of iron, and 25 to 60% of B complex vitamins. As I told earlier, meat from sheep is known as mutton, but in India, popularly, meat from goat is popularly known as mutton. But when specifically young sheep, it is referred as lamb, and meat from goat is known as chevon. So if we compare meat from sheep and goat side by side, we can see one thing very clearly. Meat from chevon has very low content of fat, that is 2.3% as compared to mutton. And when it is this result is corroborated by another study in which 5 different species of meat was observed, in which chevon had lowest content of fat. Also, it had the lowest content of saturated fat. It had the lowest content of cholesterol, and it had higher content of iron. So that's why goat meat or chevon is regarded as lean red meat, and it is the healthiest red meat among the all other species. So chevon has lower amount of saturated fatty acid along with higher content of unsaturated fatty acid, both of which are desirable. Also, it has lower amount of hypercholesteremic fatty acids, those fatty acids which are responsible for increasing the cholesterol content. Also, it has the relatively higher content of ash, which may be due to higher amount of essential minerals might be present in meat. So when we come to the detection of adulteration, various methods are there, like physical methods, which depends on the which we can know by visually, like marbling, color, texture, smell. This comes under the physical method. Next is anatomical method. Anatomical methods refers to specific structure of tissue or bones, which can differentiate one species effectively from another species. Apart from that, chemical methods are there, which include estimation of glycogen, which includes estimation of fat, iodine value of fat. This we'll discuss later. Immunological factor methods include determination of proteins or specifically immunogenic proteins which are specific to one species. And recently, one sector is growing, a research sector is going specifically in omics-based methods, which includes proteomics, genomics, metabolomics. I have kept one slide for that. And DNA-based methods includes detection of specific genes and this has been very much useful in identification of species. First, let's come to the physical methods. When we compare flavor between Chevon and mutton, Chevon flavor is comparatively rich and bold, and it is mild and delicate in case of mutton, and color of Chevon comparatively deep red, and it is tender pink to pale red in case of mutton, and due to higher fiber content in case of Chevon, it is firm and coarse, and relatively less fiber content in mutton, it appears to be tender and fine. And fat is practically absent, or marbling is practically absent between muscles of Chevon and subcutaneous tissue. Kid, but kidney of Chevon is covered with abundant amount of fat, while in case of mutton, fat is far white, colorless, and there is presence of intermuscular fat. But this intermuscular fat difference, it does not hold significance because it will vary from animal to animal and specifically based on the nutritional status. It is written that fat is absent between the muscles, but if one goat is well nourished, there are chances that intermuscular fat might be present. So these physical methods does not hold very high ground while differentiating the species. If one has very high experience, he is everyday observing, then only he can differentiate by using, but he cannot tell by 100%. So for that, we need to differentiate based on anatomical methods. So these slides are going to be very much important. So when whole carcass is there or one split carcass is there, when we can, there are chances that we can differentiate between two carcass of sheep and goat. We should go for that. First of all, we should go to scapula. Scapula of goat has a distinct neck, while it is not that much distinct in case of sheep. But another thing, important thing, scapula of sheep is comparatively short and broad as compared to goat. But one distinct feature which everyone can notice, that superior spine of scapula in case of sheep, it is bent towards back and it is thickened. In this image also you can notice it. And in case of goat, it is straight and narrow. This feature is very much distinctive. And next we should go to compare the length between radius and metacarpus. We can see radius of goat is around 2 times higher length as compared to metacarpal, while in case of sheep it is relatively slightly larger. Radius is slightly larger than metacarpal. So if we observe from the front, this image is from side, but if we observe from the front, goat thorax is flattened laterally, but in case of sheep it will appear like barrel shaped. And another feature which is very important, we should observe the sacrum. The sacrum of sheep, its lateral border is thickened and it is rolled towards inner side. You can see from this image. While in case of goat, lateral borders are thin and sharp. So this is also another very distinctive features. And if we observe a carcass from its back and wither, we can see sheep is round and well finished, while in case of goat, the back and wither region is sharp and it is little fleshed. So as we summarize, we should remember few important things. First of all, one is scapula. Other things we cannot remember, that superior spine, which is straight and narrow in case of goat, but bent back and thickened in case of sheep. It is very distinctive. Similarly, in case of sacrum, that also we should keep in mind. Lateral borders are thickened in case of sheep, while lateral borders are thin and sharp in case of goat. Similarly, if you can observe the tail region, it is fairly broad in case of sheep, and goat it is thin. And I think the biggest plus point we can find simply the radius, if we can compare the radius and metacarpal, because we can see the length of metacarpus of goat is 2 times as compared to the metacarpus. So if whole carcass is not there, then this knowledge goes to vain, because we cannot simply by physical methods, by seeing the color, by seeing the marbling, we cannot 100% justify that this is a sheep carcass or this is a goat carcass. So we should move to chemical methods. But I should tell from the beginning, chemical methods are not of great value on justifying the or authenticating the sheep. And this is may not be a very great practical value, because if we are going to glycogen content, if we are detecting or suspicion, doing some suspicion, some species might be adulterated. Specifically, if we are suspecting horse fish might be adulterated, then glycogen content may be of some value, because horse flesh has significantly higher glycogen content as compared to other animal. But if we are checking for adulteration in sausages, where there are chances that giblets might be adulterated, in that cases we can go for glycogen estimation, because liver has significantly higher content of glycogen. So presence of liver, especially pig liver, indicates adulteration of liver in sausages, because in sausages minced meat is used. By visually by seeing we cannot say. So this is there are some inherent disadvantage in glycogen estimation, because glycogen quickly depletes after death, because it is converted to lactic acid and other external factors such as nutritional factor. If animal is malnourished, definitely there is glycogen content is going to be low. And similarly, if animal is stressed, glycogen is going to deplete very quickly. So therefore, it is also not great value. So, but I have attached a method for estimation of glycogen, to which I am not going deeply. Those who want to estimate, they can take it as a reference document. Similarly, some other analysis, chemical analysis are there. That is analysis of linoleic acid content of fat. This is also useful when we are suspecting that horse flesh is adulterated, because horse flesh contains highest amount of linoleic acid, which is around 1 to 2% as compared to 0.1% in other species. And iodine value estimation of fat comes next. Iodine value estimation depends on the degree of unsaturation and saturation of the fat, because pig meat has relatively higher content of unsaturation, and horse meat also. So pig meat have relatively higher degree of iodine value. So if we are suspecting our sheep or goat meat is adulterated by horse flesh or pig flesh, in this case iodine value estimation holds a true potential. So here I have given a detailed protocol for estimation of iodine value, to which I am not going in detail, and it can be used as a reference for if anyone want to use it. Another factor is the refractive index of fat. In this case, fat is first melted and its refractive index is measured. In case of ox, sheep and goat, it is less than 40, while in case of horse and pig, it is relatively higher. Here, two parts are finished. First is anatomical methods. If it is of great value when whole carcass is present, but then we saw chemical methods are not that much useful. So, what can be the challenges if minced meat or caught meat or cooked meat is present are there? It is of great challenge, because morphological differentiation is not possible, because anatomical differences are indistinguishable. There are loss of physical properties like color, texture, and marbling pattern, and there are every chances there that there may be admixture from more than one species. If we are seeing 100 gram of minced meat of goat, there are chances that 10% will be added by maybe maybe make up by chicken, or there are every chances that 15% adulteration of sheep is there. So another thing is there, when meat is cooked, so whatever the color is also there, it is also gone. And one major problem comes in cooked meat is there is denaturation of protein, there is damage to the DNA, that is shearing of the DNA due to heat processing. So therefore those protein which are susceptible to heat, they cannot be used for identification of species. Here comes the role of bioinformatics, proteomics, by which we can identify specific domains of protein. By use of genomics, we can identify specific molecular markers which are resistant to heat. And later, these methods can be adapted to field level for detection of meat, which I am going to tell subsequently. So I have gone through some diagnostic kits which are available commercially for authentication of meat species. So this is one kit for qualitative detection of animal species content in uncooked meat and meat products by enzyme linked immunosorbent assay technique. So this is a non-competitive sandwich type immunoassay for qualitative detection. One thing to note that it is for qualitative detection, but it cannot quantify how much amount of adulterant is present in the sample. And this kit is developed for analyzing raw meat. Similarly, another kit is there which is for performing by using ELISA. It can identify the species for cooked meat. But one disadvantage is there in these kits. A set of kits are there for a separate species, so we have to perform separate ELISA for each species until we are diagnosing that which adulterant is present. And one major disadvantage is there, if we are suspecting goat is adulterated by sheep, these kits cannot differentiate. I gone through other some kits also, but sheep and goat proteins, as they are cross reacting, sheep and goat, these kits cannot differentiate. But one advantage is there, it can differentiate between cow, it can differentiate adulteration from pig, it can detect adulteration from poultry, and also it can detect adulteration from horse. But both these raw and cooked meat, they cannot detect adulteration between sheep and goat. This is one big disadvantage. So if we can equip our district diagnostic laboratories at district level, we can supply them with ELISA kits and ELISA readers. This can be a big advantage to them. But how can we differentiate between sheep and goat? This is possible by using molecular assay. I found this one molecular kit. Other kits are also available online. This one is Sure Food Animal ID 4plex for beef, sheep, goat, and another one is internal amplification control. Internal amplification control refers to one internal control DNA, which will clarify that whether our PCR is working properly or not. Using this kit, we can differentiate between whether it is a sheep meat, whether it is a goat meat. And this also, it is not applicable at field level, because and at district diagnostic level laboratories, we can equip them with PCR and we can provide them these kits so that it will be easier for them to identify which may be the adulterant. But one thing for sure, that these kits are very expensive. For limited amount of 50 or 30 number test, they are going to cost around 18,000, 20,000. But one good thing that it is very sensitive, and around 0.01% adulteration also it can detect. There are some other kits like food proof SL goat species detection kit, which can detect up to 10 genome equivalents or 10 genome copies in the analyte. And sheep species detection kit similarly there, which can detect up to 0.1 genome equivalent between sheep and goat. But these kits, they cannot be applicable at field level because we need specific instrumentation facilities. For this one kit, I found fast meat species identification kit. It requires some 10-minute processing, followed by which that meat extract, which is taken in a tube, and in that dipstick ELISA or immunostick, which is referred here, if it is deep, based on the color development, we can speculate what species it might be. It is red color in case of cow, it is yellow color in case of pig, blue color in case of poultry, orange color in case of horse. But one disadvantage is that it also cannot differentiate between sheep and goat. In case of sheep, also in case of goat, it will come green color. So how this problem of differentiation between sheep and goat can be elevated? By definitely by using research developments. One research was conducted in India, in which they developed a duplex PCR, and in which they used 3 primers, in which it can differentiate between sheep and goat. By using pure goat meat, it will detect only goat. By using pure sheep meat, it will detect only sheep, and it can detect successfully up to 10% adulteration. If both meat species will be there, like this, 2 bands will be there. So if DDLs can be equipped with PCR facility and they can be provided with these primers, it can be very useful if there is doubtful regarding sheep and goat adulteration. Also, if we are using our own primer, own chemicals, the cost is going to be very less as compared to kits. I like to highlight another study that was meat species identification using loop mediated isothermal amplification assay targeting species specific mitochondrial DNA. So LAMP PCR is another assay which holds the potential to perform PCR at ground level also, because LAMP PCR is performed at a specific temperature, unlike conventional PCR. In conventional PCR in thermocycler, we give a set of temperature and time combination, but LAMP PCR operates at a fixed temperature. So this PCR assay was developed which can simultaneously differentiate between 8 different species: cattle, pig, horse, goat, sheep, chicken, duck, and turkey. And for each species, there is unique primers. For each species, there is unique annealing temperature. So based on the which species adulteration we are suspecting, we can accordingly use this primers, and we can, by using simple DNA extraction protocols, we can pop, and by using a hot water bath also, we can perform at field level. With basic instrumentation facility, basic laboratory facility, we can perform this assay at field level. And the most importantly, the result can be visually differentiated. In case of negative, there will be no color change. In case of positive, there will be turbidity, increase in turbidity, or change in color change. There will be color change. So how LAMP PCR can be an effective substitution for PCR? No, conventional PCR, because it requires only single temperature, that is around 60 to 65 degrees Celsius, and depending on the primer used, it requires a comparatively lesser time, which is higher in case of conventional PCR. Conventional PCR requires around 1.5 hours to perform the PCR. Additionally, there will be gel electrophoresis, documentation, whole technique takes up to 2.5 hours. And the most importantly, the result can be visualized by naked eye. We do not need any specific instrumentation facility. Now, another advantage is coming by developing portable PCR. So use of portable PCR, and now we do not require classical gel documentation system and electrophoresis to analyze our PCR results. So using a portable PCR, lateral flow assay has been developed to detect the amplification using specific probes or complementary probes to the amplified PCR products. So using this portable PCR and lateral flow assay, detection of pork, horse, beef, chicken, turkey, and goat contamination in meat products have been used. So one biggest advantage of lateral flow assay is that you can see these two images. In left side image, there is 100% lamb. The first line comes for like internal amplification control. It will be giving whether our kit is working fine or not. The second line in case of 100% lamb indicates this is pure lamb. Similarly, in case of goat also, we can see two lines are there, but it can simultaneously detect many different number of species adulteration. See, in case of second strip, it could simultaneously detect 5 to 6 different species adulteration is there, and it could detect up to 0.1% meat adulterant. So unlike ELISA and unlike PCR, where we have to perform individually for each species, specifically this assay, lateral flow assay, can be developed for detection of simultaneously multiple number of species, and that too it is a potential point of care diagnostic assay. But one thing is there, we have to have a portable PCR. This is one quick method of DNA lysis, DNA extraction I have added, because if we are thinking to do thinking to take to the PCR to the ground level, we have to prepare one suitable method of DNA extraction using minimum resources. This around 500 milligram of meat is required, to which sodium hydroxide is added, and to that, which is kept at a water bath or dry bath at 75 degrees Celsius, and to neutralize its pH, we need to add 3 HCl simply. After doing this simple 20 minutes protocol, that can be used as a template DNA for PCR for meat identification, and this has been done at NRC meat, this simple DNA extraction protocol. And now, identification of immunodominant proteins has been a great use. This work was developed at NRC meat, and it is a very potential assay developed, as I can say, because it is a point of care assay. We do not need to do any PCR anything, and it can detect proteins from raw, heat processed, commercial samples, even in meat offals, using simple 10 minutes extraction protocol. It uses gold nanoparticle levels, and immunoglobulins for meat species identification. And it involves a quick 15 minutes procedure, and it can detect up to 0.063% pork and chicken and 0.125% lamb. So such type of kits can be provided directly to the dispensaries. Of course, we have to initially standardize, and these kits can be prepared at biological products division of the state, and simply this can be supplied at the ground level. When a veterinarian will be in need, he can simply extract this meat proteins and simply 1 to 2 drop adding the sample. He can simply tell whether it is a pork, it is a chicken, or it is a lamb adulteration. So now comes the omics based method. So this comes the pure research thing. It involves several types of omics like proteomics, which involves identification of specific proteins to a species. Genomics involves identification of specific genes. Metabolomics involves identification of specific metabolites, and transcriptomics involves identification of specific transcripts or mRNA. Similarly, lipidomics involves identification of specific lipids or fatty acid. Elemental profiling involves identification of various type of elements, and isotopolomics involves identification of various types of isotopes. Among these, proteomics and genomics holds great value in species authentication purpose. And for geographical authentication, elemental profiling and isotopolomics. See, in this omics based method, we can go beyond the traditional species authentication, because now there is rising demand for meat from organically raised animals. There is rising demand from meat from specific breed, like some has specific intent to consume black Bengal breeds meat because of its specific taste. So up to these also we can authenticate using these advanced techniques. Similarly, breed authentication is also by genomics we can use. And recently, adulteration with giblets, one work was conducted at NRC meat. In that, they used microRNA markers. Using that, in minced meat, they could detect adulteration from giblets. Of course, it was droplet digital PCR was used. It is a very high-end PCR machine, but these techniques can be taken to ground level. Once the specific molecular markers are detected, then that PCR LFA, these techniques which can be used at ground level. And similarly, detection of giblets in goats and sheep also can be done. And various techniques which are used for analysis of this omics based method includes gas chromatography, liquid chromatography, nuclear magnetic resonance spectroscopy, and matrix assisted laser disruption ionization, time of flight mass spectrometry. These are the techniques which are used for these omics based studies. So if you are summarizing, if our whole thing, if a whole carcass is available, then veterinarians first should go for the anatomical features. Then anatomical features can make it is a very easy and quick method by which they can differentiate the species. Similarly, we should supply either the dipstick kits which I showed earlier or lateral flow immunoassay. By supplying these kits, it will be very easy for a veterinarian to identify what is the species. Even now, kits are developed which can identify from cooked meat. Similarly, among the PCR techniques, lamb PCR is now emerging as a point of care diagnostic assay. It has the potential. Similarly, PCR and lateral flow assay based kits also can be used with a minimum instrumentation facilities. But in the state or regional level, we should have a high-end authentication laboratory which can authenticate the breed, which can authenticate the rearing, whether our sheep was reared using or goat was reared using organically. This authentication gives a great satisfaction to the consumer. So, sir, this is all I have to say.